Okay, welcome to the first lab. I hope you enjoyed it, and here's some of the answers for the questions in the lab, where we're exploring the database, the NCBI GenBank database, as well as doing our first blast. So questions 1 to 5 in the lab explore the power of limits, so we can use very specific terms to narrow down our search in GQuery. We can also use Boolean operators like and or not to really specify the exact records or small subset of records that we're interested in. Of course, the most specific way of accessing a record is using the accession number, such as the one we're working with in the lab today, NP00131838. And in that case, we just get one record returned for the protein. So if we go to that record and we start exploring the record, we see lots of hyperlinks. It looks kind of messy, but in fact it's very well organized and structured. And if, for instance, we click on the source organism hyperlink, we see lots of information about the source organism, namely Arabidopsis thaliana, which is kind of like the fruit fly of the plant world. Grows very quickly from seed to seed, 6 weeks. And here, for instance, we can read also the taxonomic lineage of the organism from the NCBI taxonomy browser. You can see that it's eukaryotes, Viridiplantae, and so on, all the way down to the genus Arabidopsis. We can also see that there is, in question 6b, asks, has the genome of this organism been sequenced? Is there a genome project? And the answer is yes. So if we click here, we see that there is a genome link. And if we click on that, we can see the information about the genome project. And in fact, the chromosome sequences are listed, and you can tell that they're chromosome sequences by the RefSeq identifier starting with NC. C is contig or chromosome. And if we click on that one, which is NC underscore 003070.9, we're actually taken to the entire sequence of chromosome 1. All right. When we go back to the GenBank record and click on the CDS link, the question 7a is asking where did this take you, or what happened when you did this. And when we click on the CDS link for that NP record, actually the CDS, the coding sequence itself within the record, is highlighted in a brown color background here. The next part of the lab deals with exploring related information, and there's a bunch of linked information for each record in GenBank. We can move to the gene record, so the nucleotide sequence information for the protein record, the corresponding nucleotide sequence information. And this is useful for understanding where a gene is located in a chromosome, what its chromosomal neighbors are, those kinds of things. And we can get the gene's location in the chromosome by simply hovering over this pictograph here, and we see a pop-up that comes up, and we see that the gene's location is from 12,368,220 to 12,370,420. So that's 2,200 nucleotides long. Question 9b asks how many exons do we see for this gene, and our gene is pub12, and we can simply count the number of green boxes. So 1, 2, 3, 4. So there are 4 exons for this gene, and the things in between those, here, here, and here, are introns. Question 9c asks, what are the names of the genes surrounding it? What is the genomic context for the pub12 gene? So we can look just a little bit up the page, and we see here is pub12. We see that there's another gene just to the 5' side of the pub12 gene, and that's AT2G28815. There's another gene going in the opposite direction as pub12 on the top strand, actually, called AT2G28810. And then there's another stream on the 3' side of pub12 called XBAT31. Question 9D asks, does pub12 have any conserved domains? And if we use the conserved domain related information link on the right side of the page, we can click on that, and then we're taken to some conserved domains for the pub12 gene. And the conserved domains, so it's a database of domains which are important functionally for the functioning of a protein. And we see that there's an armadillo beta-catenin-like repeat, as well as a ring finger motif within the pub12 protein. So question 9E asks, what is the biological process that this gene is involved in? And we're talking now about gene ontology terms. Gene ontology is a system for classifying the function of proteins, and it's species agnostic. A gene from any species can be slotted into the controlled vocabulary of gene ontology, which has 3 main parts: function, process, and subcellular localization. This particular question is asking about the biological process. So if we go back to the gene page and scroll down, we can actually see that the gene ontology biological process is defense response to bacterium. All right, we can also explore some additional links which are provided by external resource providers. So if we go to the bottom of the page and click on the expand option for the gene link outs, you see that there are a bunch of links to external resources. So Gene Visible contains gene expression information. BioGPS is kind of an overarching consolidation website. OrthoDB will give you orthologs for that gene, those kinds of things. Question 10b asks, why is the length of the mRNA different from the value you can calculate from the start and stop positions in question 9a? So there, if we calculate that value, so that's here are the start and stop positions here. That value is 2,200. If we're looking at the related information in terms of the RefSeq RNA, which we're doing in question 10b, we see that the length is only 1,949. And this has to do with the central dogma of DNA. In the case of NM0013369190, what has happened is that the gene has been transcribed from the genome, from the chromosome, and then the introns have been spliced out. So Sharp and Roberts got a Nobel Prize in 1977 for interrupted genes, and when there's transcription, those introns get spliced out to create an mRNA, to create a transcript, which then gets translated into the protein. So we'll explore this actually in the next section when we start to do some blasting. All right, in part B of the lab, we're exploring again the mRNA, and that's the exact same record that we just looked at, which was linked to our protein by the RefSeq by the RefSeq link. And here we see that there's some different parts in that gene. So we've got the gene and the CDS. And if we actually go back to the genomic record, we see different parts of that genomic sequence belonging to the composing the mRNA and the CDS features. So the gene goes from 1 to 2,201, and the mRNA feature corresponds to positions 1 to 1296, 1383 to 1832, 1916 to 2032, and 2116 to 2202. And the reason for this is that what's happening, as I mentioned before in the previous slide, is that these introns are getting spliced out. Then you'll notice that the CDS feature actually corresponds, it starts at position 169 and ends at 2169, not 2201. And the reason for this has to do with these two regions at either ends of the mRNA. These are the 5' untranslated region and the 3' untranslated region. So when a transcript gets produced, that would be the thing in purple here, the translation does not start at the first nucleotide of the transcript, at the first position. It starts actually a little bit downstream, so 3' from the start of the transcript, and it starts just after the Cossack sequence. So there's a recognition sequence here that the ribosome binds to, and translation will start typically at the first ATG, which encodes for methionine. Conversely, at the 3' end of the transcript, there's a stop codon. That's where translation will stop, but the translation typically doesn't go right to the end of the transcript. It ends a little bit upstream of the end of the transcript, right at the last stop codon. And so that's why there's a difference, and it's important for you to recognize that between the gene that's on the chromosome versus the mRNA, which contains both 5' and 3' UTRs typically, versus the coding sequence, which are the nucleotides that are actually translated into proteins. So that's something to keep in mind when you're looking at GenBank records. All right. When would you want to use Megablast? So here we're going to start blasting. So Megablast is very useful for searching, say, a set of sequences from a shotgun sequencing project against the reference genome. You're looking for highly similar matches with only a few mismatches here and there. Discontinuous Megablast would allow you to search for sequences that are highly similar but might have a few small insertions or deletions. And of course, today we're actually using BLAST-N, which is sort of the general purpose searching for more distantly related sequences. And what we're looking at in question 2 is what is the expect threshold? So the expect threshold is the value below. So if sequences have an e-value that is better, that is to say smaller than that expect threshold, then those sequences will be returned in the BLAST search. And we talk a little bit more about e-value in next week's class lecture. And if we decrease it, if we make that e-value smaller, say setting it 1 × 10⁻80, which is one of the quiz questions, then basically sequences only with that e-value or better will be returned in our BLAST search. Another thing that you can adjust here is the maximum number of target sequences. So if the maximum number of target sequences is reached, it becomes 100, and those sequences have an e-value that's better than the cutoff, then you will only get 100 sequences. You can't get more than 100 sequences. This value overrides this threshold value. What would be the effect of increasing the word size? So if we increase the word size, and we'll talk a little bit more about how BLAST works next week, we would increase the specificity of the search. Why is there a low complexity regions filter? What should we keep it on? So oftentimes in biological sequences, we have stretches of poly A's or poly T's, or in the case of proteins, polyproline stretches. And those aren't particularly biologically informative. They occur sort of randomly throughout genes in the genome. So we might want to mask those out when we do our search, and typically it's a good idea to leave that checkbox on. So just as a clue for your quiz question, quiz 2, you should keep all the settings the same as in the lab manual, so figures 14 and 15. All right. Okay, question 3a asks how many sequences are in the NR database. And if we actually click on the search summary, we will see some information about the database size. So number of letters is that very large number there, and number of sequences is 45 million. Question 3b asks which sequences are not included in the NR database, and this is a bit of a trick question. You actually have to go back to the BLAST input page and click on the question mark beside the database name, and here you'll see a description of that particular database, the one that's selected. It says nucleotide collection consists of GenBank, AMBL, DNA database of Japan, PDB and RefSeq sequences, but excludes these sequences, including genome survey sequences, whole genome shotgun sequences, some other ones. So just keep that in mind when you are searching. You don't actually search every single sequence that is in GenBank. These colored bars will actually see this information depending on which line we're mousing over. So for the first one, if we mouse over this first bar here, the genus and species of the best hit is Arabidopsis thaliana, and then everything else that's not quite as good a match is ordered below the Arabidopsis thaliana hit that we found, and that's the sequence that we actually use to search, so that should be a perfect match. If we click on one of the entries, we're taken to the record further down the page where we can see the exact alignment and how that looks. So there are 3i, how many sequence matches are listed for this query sequence? So there will be 100 sequence matches because that's what we specified in the input page. That's the default, 100 sequences with 121 blast hits. So sequence can have actually can match in multiple regions, and each region that matches is counted as a blast hit. So you could have 3 or 4 hits per sequence. So what happens if you click the accession hot link in the detail table summary? You will be taken to the GenBank page for that particular sequence match, and if you click on the alignments link, you'll go to the alignment section, which looks like this here. Question 3L asks, what do the vertical bars represent between the query sequence, which is the sequence along the top, and the subject sequence, which is your match in the database? So if there's a vertical bar that looks like this, that means that the nucleotides at a given position are the same on both the query and the subject. And question 3M asks, what does strand plus plus or strand plus minus mean? And this refers to the anti-parallel nature of DNA. So keep in mind that when we look at a piece of DNA, we can actually read off sequence on the top strand, on the Watson strand in this direction, and we can actually read off other sequences on the bottom strand, on the Crick strand, going in the opposite direction. So this end is 5' to 3', this strand is 5' to 3', and this strand is 5' to 3' on the opposite side. So pub12 is actually on the Crick strand in the case of Arabidopsis chromosome 2. But say we're searching against the sequence database, the sequence could be chromosomal sequence could have a match on the other strand. So BLAST will actually search in both directions, and that's what the plus plus and the plus minus refer to, depending on which strand the match is on. So question 3N asks, so we're looking at a reformatted BLAST output, and here these are query anchored with dots for identities. And if we reformat and look at the output, we see instead of these pairwise alignments, we actually see the query sequence here. A dot denotes that the position, the nucleotide at that position is exactly the same as in the query sequence. And here we start to see a few changes in these other sequences. So this T, for instance, has been changed to a C. This G has been changed to a T in all of those related sequences. And this kind of weird display here, representation here, denotes that an A has been inserted in this sequence. And this is kind of like a poor man's version of a multiple sequence alignment. We can't really represent insertions with gaps in this particular representation, and the way they NCBI's or the BLAST programmers have decided to represent insertions is with this depiction here. All right, so what we're doing now is we're returning to the original format and going back to pairwise format, and now we're looking at the graphical summary and asking whether or not there are any lower scoring segments. And in fact, for this particular record, we do see a couple of parts that don't have as good a bit score. So the bit score here would be around in the 50 to 80 range, and then the bit score down here would be in the 80 to 200 range. And so actually, if we mouse over that little bit, we can see that the E value is actually is not very good. So it's 0.0046 versus for something like this, we saw that the E value for the first sequence was 0.00, which is the best it can be. And question 3P asks, do you think that these sequences are nonetheless homologous? And I would answer yes. So even though if we go to that little region and we click on it, we see that the number of identities is 71 out of 98, so around 72%. So it's pretty high. But in fact, BLAST has given a fairly poor E value. And the reason for this, as we'll see next week, is that the way BLAST calculates the E value also takes the length of the match into account. So this region just has a very short stretch of match, and that's a reason for the not very good E value. But nonetheless, in the context of the entire sequence, from this end to this end, we see very good match with bit scores above 200 for most of the rest of the sequence. So I would say that those sequences are homologous, and that's what we're looking for. A good stretch of match across the entire sequence to call something homologous. So that's it. By the end of Lab 1, you should know how to search for records at NCBI using both search terms and identifiers and the GQuery interface, or by using a nucleotide sequence and using BLAST. You should also know the difference between a GenBank accession number, a version number, and a GI number. You should understand the difference between the nucleotide sequence database part of GenBank and the protein sequence part of it. So often the protein sequences are derived from the nucleotide sequence records. You should also know the parts of the GenBank record and be able to switch between sequence formats. You should easily be able to get a FASTA formatted sequence out of the database. You should be familiar with the interconnectedness of various NCBI databases and be able to call up linked records with ease. Just click on them basically. You should be able to use nucleotide BLAST-N to search GenBank, and you should also be able to interpret the output. What does the E value tell you? What is a good E value? You should understand the meaning of homologous, orthologous, and paralogous sequences. You should also be able to use the help function to address any questions that you might have with regards to the NCBI interface. All right, and see you next week. Thanks a lot. All right, that's it for week 1. So I think you will have seen how we can filter our results in NCBI Search to identify the sequences that we're interested in, and also how we can use BLAST-N to identify similar sequences in the database. So next week, we'll continue exploring GenBank using BLAST-P this time, which is Protein BLAST. And the other BLAST tool that we'll use is something called SciBLAST. So SciBLAST is very cool because we don't have to use a default similarity matrix, which we'll talk about in next week's lecture. Instead, we can actually generate a custom matrix, and this helps us to identify more distantly related sequences if we can't find anything that's closely related. So that's it for this week, and I hope to see you next time. Thanks. Bye.